Your muscles may be doing something far more important than moving your body. They may be sending signals that help the brain control aging. A 2026 study discovered a communication circuit connecting skeletal muscle, the hypothalamus, and brown fat. As mice grew older, this circuit weakened. Their muscles released less of a protein called Mimecan. Neurons in the brain lost part of their ability to receive the signal. Brown fat became less active, and control of body temperature declined. Then researchers restored Mimecan in old mice. The communication circuit became more active, and the animals lived longer. This does not mean Mimecan is a human longevity treatment. The experiments were performed in mice, and scientists do not yet know whether increasing this protein would be safe or effective in people. But the study reveals a surprising way to think about aging. Your organs do not age independently. They age partly because they stop communicating as clearly as they once did. Skeletal muscle is often described as the tissue that allows us to walk, lift, breathe, and maintain posture. But muscle is also an endocrine organ. It produces molecules that travel through the blood and influence distant tissues. These muscle-derived signals are sometimes called myokines. Some help regulate inflammation. Others influence glucose metabolism, fat tissue, bone, the immune system, and the brain. During exercise, muscle does not simply consume energy. It sends messages telling the rest of the body that energy demand has changed. The new research suggests that one of these messages may help maintain a communication pathway involved in longevity. The protein is Mimecan, also known as osteoglycin. Researchers did not begin by assuming Mimecan controlled lifespan. They discovered it while comparing gene activity in young and old skeletal muscle. Mimecan production was lower in older muscle. The team then asked whether that decline was only another sign of aging or whether it actually changed how the body functioned. They genetically altered mice so their skeletal muscles produced less Mimecan. The result was not limited to muscle. The animals had reduced activity in brown adipose tissue, commonly called brown fat. Their ability to regulate core body temperature weakened. This was the first major clue. Muscle appeared to be controlling fat tissue located elsewhere in the body. But it was not communicating with brown fat directly. The signal first traveled to the brain. Mimecan entered the circulation and acted on specific neurons in the hypothalamus. The hypothalamus is a small brain region that helps regulate temperature, hunger, thirst, sleep, hormone release, metabolism, and other functions required for survival. The neurons involved in this study carried melanocortin-4 receptors, or MC4R. MC4R is already known to play an important role in energy balance. Genetic disruptions affecting this receptor can cause severe obesity in humans because the brain loses part of its ability to regulate appetite and energy expenditure. In the new study, MC4R-positive neurons helped control the activity of brown fat. Brown fat is different from the white fat stored around the waist and internal organs. White fat primarily stores excess energy. Brown fat burns fuel to generate heat. Its cells contain large numbers of mitochondria and a protein called UCP1. Instead of using all available energy to produce cellular fuel, UCP1 allows some of that energy to be released as heat. This process is called non-shivering thermogenesis. It helps mammals maintain body temperature in cold environments without relying entirely on muscle shivering. Brown fat also affects glucose and lipid metabolism. People with more active brown fat often show healthier metabolic profiles, although scientists are still determining whether activating it can safely treat obesity or diabetes. Brown fat activity tends to decline with age. The new study suggests that part of this decline may begin outside the fat tissue itself. The problem may start in muscle, travel through the blood, reach the brain, and then weaken the nerve signals sent to brown fat. The circuit works like a three-part relay. Muscle releases Mimecan. Mimecan activates specific hypothalamic neurons. Those neurons increase sympathetic nervous system signals directed toward brown fat. The sympathetic nervous system is part of the body's automatic control network. It can release signals that tell brown fat to increase fuel burning and heat production. But the study uncovered another layer. Mimecan did not simply attach to neurons and turn them on. It helped maintain structures called primary cilia. A primary cilium is a tiny projection extending from the surface of many cells. It behaves somewhat like an antenna. It helps the cell detect chemical and mechanical information from its environment. In the relevant hypothalamic neurons, these cilia helped maintain normal signaling. As mice aged and Mimecan levels declined, the cilia became shorter. The neurons then became less responsive. Sympathetic stimulation of brown fat weakened, and temperature regulation deteriorated. Imagine a radio tower gradually losing the length of its antenna. The message may still be transmitted, but the receiver can no longer detect it clearly. The aging problem was not only that muscle produced less of the signal. The brain's receiving equipment was also deteriorating. To test whether Mimecan truly controlled the pathway, researchers used both loss-of-function and gain-of-function experiments. When they reduced Mimecan production specifically in skeletal muscle, brown fat function and temperature regulation worsened. When they increased Mimecan, the pathway became stronger. The researchers then reduced MC4R inside the relevant hypothalamic regions. This eliminated the beneficial effects of Mimecan overproduction on brown fat. That experiment was important because it showed that Mimecan was not simply activating brown fat through some unrelated pathway. The brain receptor was necessary. Finally, the researchers restored circulating Mimecan levels in older mice to levels more similar to those seen in younger animals. The treatment significantly extended lifespan. This was not merely a short-term rise in body temperature. Restoring one declining signal altered the survival of the animals. However, the study does not establish exactly why the mice lived longer. Improved brown fat activity may have supported temperature control, energy metabolism, glucose regulation, circulation, or the ability to respond to environmental stress. Mimecan may also have produced effects in tissues that were not fully examined. Longer life can emerge from several connected changes rather than one isolated mechanism. Core body temperature itself has an unusual relationship with aging. In mammals, the ability to maintain stable temperature often weakens later in life. Older people are more vulnerable to cold, partly because of changes in circulation, muscle, fat distribution, metabolism, and nervous system control. But a higher temperature is not automatically healthier. Fever is dangerous when excessive, and chronic changes in temperature regulation can reflect disease. The goal of the Mimecan circuit is not simply to make the body hotter. It appears to preserve the ability to regulate temperature appropriately. That difference matters. A healthy system responds when heat production is needed and reduces activity when it is not. Aging often reduces this flexibility. The body becomes less capable of adjusting to changing conditions. The study also helps explain why maintaining muscle may influence much more than strength. Loss of skeletal muscle with age is called sarcopenia. It increases the risk of falls, disability, poor recovery, and loss of independence. But if muscle is also a communication organ, losing it may reduce important signals reaching the brain, fat, immune system, and other tissues. An older person can therefore lose more than physical power when muscle declines. They may lose part of an internal information network. This does not prove that exercise increases Mimecan in humans or that lifting weights activates the exact longevity pathway found in the mice. The researchers identified Mimecan by comparing young and aged muscle, not by running a human exercise trial. People should not assume that a particular workout, food, cold shower, or supplement will reproduce the treatment. No approved Mimecan drug exists for slowing human aging. The protein could also have different effects depending on dose, tissue, age, health condition, and treatment duration. A signal that is beneficial when restored to a normal level could become harmful when pushed far above that level. The MC4R system also affects appetite and body weight. Interfering with it could produce unintended metabolic or neurological consequences. Translation from mice to humans is especially uncertain in brown fat research. Mice have much more active brown fat relative to body size and depend heavily on it for temperature control. Laboratory mice are commonly housed at temperatures below their thermoneutral zone, meaning their bodies must continuously generate extra heat. Humans usually wear clothing, heat their homes, and experience different environmental conditions. A treatment that produces a major effect in laboratory mice may have a smaller or different effect in people. The lifespan experiment also does not tell us whether Mimecan extended healthy, functional life or simply delayed death. Researchers need to examine mobility, cognition, frailty, disease burden, and other measures of health span. They must also test the treatment in different sexes, genetic backgrounds, and environmental conditions. Still, the research offers an important conceptual shift. Aging is often studied as damage building inside individual organs. DNA mutations accumulate in cells. Mitochondria lose efficiency. Proteins become damaged. Senescent cells release inflammatory signals. All of these processes matter. But even healthy organs cannot coordinate the body if the messages between them disappear. A muscle may still contract. A neuron may still survive. Brown fat may still contain functioning mitochondria. Yet if the communication chain connecting them becomes too weak, the system fails as a whole. Aging may therefore involve not only damaged components but damaged relationships between components. This creates a new class of possible treatments. Instead of replacing entire organs or targeting one damaged cell type, scientists may try to restore the signals that coordinate multiple tissues. The challenge will be identifying which communication failures truly drive aging and which are only consequences of it. Mimecan is unlikely to be the only signal involved. Muscle releases many molecules. Brown fat communicates with the liver, heart, immune system, and brain. The hypothalamus receives information about nutrients, hormones, temperature, stress, and activity. The body is less like a collection of machines and more like a conversation involving billions of participants. The new study identified one sentence in that conversation. With age, the sentence becomes quieter. When researchers made it audible again, old mice lived longer. For people today, the practical message remains familiar even if the mechanism is new. Preserving muscle through appropriate resistance exercise, regular movement, sufficient nutrition, and treatment of chronic illness remains important for strength, metabolism, and independence. But this research suggests another reason muscle matters. It may help keep distant organs connected. The deepest effect of aging may not be that the body forgets how to function. It may be that its tissues gradually stop hearing one another.